Greetings from askIITians. My name is Vasanth. I'm a biology faculty in askIITians. Today we're going to discuss about a grade 12 chapter, which is molecular base of inheritance. The chapter's weightage with respect to NCERT is pretty less than 12 or 13 percentage. But when it comes to NEET, it is combined with the other biotechnology and genetics based chapters, and it is a whopping 24% of the weightage to the NEET examination. So today we're going to discuss about one of the few topics from this chapter. Come on guys, let's get into the video. So today we're going to discuss about central dogma, a part of central dogma. So central dogma includes transcription and translation. So transcription is a process where the genetic material, which is a gene, is being transcripted into an RNA sequence. Here we need to know the central structure of a gene. Here a gene actually has a promoter site in which an RNA polymerase will get attached to, and then it will create a script or RNA script from the template strand, and it has to stop at a moment because it cannot keep on going. Then the mRNA script will not be available for us. So once after it reaches the terminator site, the RNA polymerase will get relieved out of the gene sequence. So once we will get an RNA transcript. The RNA transcript will be containing both exons and introns, and that has to be modified, and that process is called splicing. So let's see them part by part, by stage. So first here you can see the RNA polymerase is ready to get attached to the promoter region. So promoter region will have a definitive sequence which will be identified by the RNA polymerase, and then the RNA polymerase will attach itself to the promoter site. So once after the RNA polymerase gets attached into the promoter site, it will start to arrange or align the nucleotide sequences of RNA polymer, and then it forms the mRNA. So in this we can see the RNA polymerase adds the nucleotides to create an mRNA sequence from a DNA template. Okay, and here once after the mRNA is synthesized, the mRNA will be found to be complementary to the base pairs that are found in the RNA in the DNA in the template region. And once after that happens, this is an elongation stage. There is a termination stage where you can see the RNA polymerase reaches the termination site, and it will be relieved out of its position, and then or released out of its position, and then the mRNA will be available free in the nuclear medium. So once after this happens, the next step is where the non-coding regions of the mRNA has to be spliced out. So we call the non-coding regions as introns, and introns are spliced out. We call them RNA splicing unit. So after the splicing takes place, the mRNA will be complete only with the coding regions, which will be very much available for translation. Translation is a process which involves protein synthesis from the mRNA, which has been formed from the DNA template and modified by RNA splicing method. And that mRNA, which has been spliced, will move out of the nucleus through the nuclear core and then available in the cytoplasm for translation process. So today guys, I hope you understood and you enjoyed the video with me in learning the central dogma, a part of the central dogma which involves transcription process. To learn more and to enjoy more videos, subscribe to askIITian classes online. Thank you guys. Thank you so much.